Hello, and welcome to Mayer's Teaching Institute. Today, we will be covering up a chapter from the Maharashtra State Board of Secondary and Higher Secondary Education syllabi, and particularly for Class 12th Higher Secondary Education: Human Health and Disease. So, in this presentation or video, we will be covering up the following topics. We will not be specifying into certain diseases like malaria, dengue, etc. But we will understand the terms involved, the concepts of immunity, the cells that are working for our immune system, and the whole interrelation with antigens and antibodies. So, thus, in order to start this topic, we should understand that health is not just the absence of disease or a physical fitness, but according to World Health Organization, it is a state of complete physical, mental, and social well-being, and not just the absence of disease. Thus, we are only able to prevent ourselves from any kind of foreign bodies, the foreign bodies being the antigens which evade or stimulate our immune response. And this is done by the immune system of our body. The immune system of our body works in resisting any damage within our body or any disease causing agent through its defense mechanisms. Thus, immunology is the branch of science which deals with the study of this immune system and its relation with the foreign bodies and their roles in resisting infections by various pathogens within us. And who is the one who does all this work? They are none other than the antibodies. So, thus, we'll move on to classify the immune system. The immune system is innate or acquired. An innate immune system is the resistance to infections that an individual possesses by virtue of his or her genetic makeup. It is natural or rather inborn and is a defense mechanism of the body. It is basically something that we get from our parents, something that is already within us, which is why when a child is born, there has to be certain amount of immunity already within that child. However, the rest of the immunity the child will acquire as he or she grows. That is the reason why many a times we have found that children fall sick very easily when they are in their small or infant age. But however, certain diseases their body just cannot resist, as a result of which, as we grow, certain vaccinations involving TB and involving polio drops, involving the, let's say, for example, the tetanus injections, that we have to instill these kind of medication into the child so that the body develops its immune system, so that when the person is put into a situation, the person is ready, the child is ready to face that kind of a disease causing agent. And that is what is about innate immunity. The first thing in our very own body as children is the epithelial surface that makes maximum protection. The intact skin or mucus covering the body protects it considerably against the invasion by microorganisms. The healthy skin possesses bactericidal activity due to the presence of high concentrations of salts by the drying sweat. So, thus, you realize that what kind of responses our body can make as a child in preventing certain diseases. For example, let's say there's someone who coughs or sneezes near a child. That doesn't necessarily mean that the child will get a cold or cough immediately. The child's innate immunity will try to resist the viruses that are causing a cold or a cough. However, sometimes some children are born with very weakened immunities. They may be susceptible to such conditions, but that doesn't mean it is the case with all children. Antimicrobial substances present in the blood. The It is basically the presence of the more than 30 serum proteins circulating in our blood in an inactive state. The presence of microbial pathogens activates the complement cascade to eliminate pathogens. So basically, this is where our body's self-immune system, which is present within our blood or within the liquid systems of our body, come into action, and they will take care of the body. Coming next, cellular factors in innate immunity. Natural defense against the invasion of blood and tissues by microorganisms and other foreign particles is mediated to a large extent by phagocytic cells, which ingest and destroy them. Phagocytosis is a case of destruction of cells. So the body itself, its own cells, will perform certain action that will capture the pathogen or the antigen or the foreign body and destroy it, as a result of which the body is prevented from facing any kind of damage or disease arising from that organism. A class of lymphocytes called the natural killer cells are important in non-specific defense against viral infections and tumors as a part of the innate system of cellular factors. Fever. Increase in body temperature following the infection is a natural defense mechanism. It helps us understand that we are facing some kind of a disease or some kind of an infection within our body. Then the acute phase proteins. Infection on injury leads to a sudden increase in the concentration of certain plasma proteins, collectively called as acute phase proteins. Now, these proteins in our body are all going to, sort of prepare ourselves to uh, protect us from certain kinds of disease causing agents that we may be exposed to within a short span of time and prior to the entire overall immunity system development. Next, coming to acquired immunity. The resistance that an individual acquires during the lifespan is known as acquired immunity. It is also called as adaptive or specific immunity. It involves the formation of the antibodies in the body which can neutralize an antigen. This is very, very important. In the current world with the coronavirus, our body is not able to synthesize a suitable antibody, and as a result of which, there is the quest for a vaccine. There is the quest for a medicine that can help us create such kind of an antibody to toss out the virus from our body. But those who can manage to make it, synthesize it within time, they are surviving, they are recovering from the situation. But there are many people whose systems, immunity systems, are not functioning well enough, particularly children below 10 and children, olders who are senior citizens above the age of 60 and so on. Their immune systems are weakened, as a result of which they tend to struggle to create these kind of antibodies to protect themselves. The acquired immunity can be two types: active immunity or the passive immunity. Active immunity, it is a resistance developed by individual as a result of an antigenic stimulus, also known as the adaptive immunity. Whereas the passive immunity is the immunity acquired when a ready-made antibody are received by the body cells. That is, the body does not take any active part in the production of immunity. Basically, when we give a person an antibiotic to prepare them, to help their immunity, sort of fight against the antigen that's coming in, it is passive. But when your own body can do it naturally, it is the active immunity. Thus, active immunity is also classified in two ways: natural and artificial. Natural immunity is the one where your body is faced to the infection. You develop a resistance to that infection by developing your own antibodies and eventually be able to prevent yourself from suffering it for a lifetime. One such example is a person who has recovered from an attack of measles develops a natural acquired immunity, which is an active immunity, and that is for a lifetime. On the other hand, an artificial acquired active immunity, it is the resistance induced by vaccinations. During the vaccine, the body has been stimulated to form antibodies by the own immune system. So basically, in polio or BCG vaccines, we are introducing sort of dummies of the pathogen. So your body is now going to fight them by preparing its own army of antibodies. And when that antibodies army is ready, and we are actually faced with a real life situation where we are exposed to such kind of bacterial or viral causing pathogens, we are able to tackle them without too much difficulty. Then moving on, next is the natural and the artificial acquired passive immunity. The natural: before birth, maternal antibodies are transferred from the mother to the fetus. After birth, antibodies are transferred from the mother to the infant through colostrum, the first milk of the mother, and is continued throughout the period of breastfeeding. The antibodies received by the baby from the mother remain in the body for a short span of time And this is called as natural acquired passive immunity, which remains for a very, very short span of time. However, the artificially acquired passive immunity, the immunity which is developed by injecting previously prepared antibodies using serum from humans or animals. For example, antibodies obtained from hyperimmunized horses are injected to humans against rabies pathogens, but these are short lived. Another case of example is the H1N1 virus. Once you get the virus, you're supposed to take the vaccination. But however, you have to repetitively take this vaccination after every year, because the effect of these kind of vaccinations lasts only for a year, and these become a part of the artificial passive acquired immunity. Therefore, the acquired immunity or the adaptive immunity has certain specific unique features. First of all, it is about its specificity. We can produce antibodies or T lymphocytes against a particular antigen or a pathogen. It's not that we have only one set that can treat only a certain class of such pathogens, but we can create, our body has a very wonderful system that can actually create the pathogen and try to tackle this pathogen without any trouble. But it can be done for a specific pathogen. Diversity, it can recognize different pathogens, different foreign molecules, so that makes it completely different. And further on, it is able to discriminate between the own body cells and the foreign bodies, which means it's your own immunity is not going to fight against your own self. There have been cases when that happens. I would not want to move down that lane, it's a whole topic by itself. But yes, being glad about this, our majority of the times our body's immune system is able to decipher between a foreign body, our own body, and even to classify between different other pathogens or foreign bodies. Now, once our body faces the virus or the bacteria or the pathogen, we are able to create a memory system. Basically, if we are encountering that virus or the bacteria or the pathogen again, our immune system is prepared. We will be able to give it a quicker, stronger response and thus suffer less. This is when I spoke about the same example on measles, that once we have developed this immunity against measles, it's for a lifetime, which means our immune system is prepared. It's got it in its memory. It has understood the mechanism and it is prepared if we are ever faced with measles again to toss it out. Continuing next, what are the various cells of the immune system? There are two types of cells, the lymphocytes and the antigen presenting cells. Antigen presenting cells, also known as the APCs. Now, the lymphocytes. There are two types of lymphocytes. First of all, the two lymphocytes are both obtained as a part of the blood corpuscles, which come from the stem cells. And these corpuscles undergo a differentiation in the gut. Then it is called as the bursal or the B lymphocytes. And otherwise, if it's differentiated in the thymus gland, then they are termed as T lymphocytes. Now, these lymphocytes form the main component or the main cells of our immune system. Now, the B lymphocytes form the humoral or the antibody mediated immune system, while the T lymphocytes form the cell mediated immune system. Both the immune systems need an antigen to come into action. Only when the antigen is present can they start behaving in a particular way. But however, there are different ways in which they can respond, and we will specify on their activity in time. Now, the antigen presenting cells. The antigen presenting cells would engulf or invade a pathogen and process the antigen. And then this processed antigen is presented to the helper T cell, who will do the remainder part of the work of destroying the cell and tossing it out of the body. For that, we will need to understand how our T lymphocytes work, and then only we'll be able to understand that why does the antigen presenting cells need to actually engulf a pathogen and bring it to our Mr. T lymphocytes. So the mechanism is something like this. The T lymphocytes, when it comes in contact with an antigen, it will form 4 clones of these T cells. Though these 4 look similar, but they all have different functions. So therefore, the 4 are the helper T cells, the killer T cells or cytotoxic cells, suppressor T cells, and the memory T cells. The helper T cells will perform several types of functions like the proliferation of the T cells, stimulation of the B lymphocytes. So basically, it is helping to prepare more lymphocytes for any kind of further action. Means it's trying to prepare the army to become bigger if needed, to toss more amount of antigens out of the body. Also, it is stimulating the B lymphocytes to be ready for futuristic responses. The killer T cells or the cytotoxic T cells, they directly attack and destroy the microbe. These killer T cells will bind to the infecting cell, bind the infected cell, basically the R cells in the body, it will directly bind to that, secrete perforins. These perforins will make a hole into that cell, release the substances that can kill the foreign bacterial, and these foreign materials are cytotoxic T cell. The suppressor T cell, on the other hand, will help prevent the entire immune system against attack on the own body cells. So sometimes our own immune system can go and attack all the other cells of our body. So the suppressor T cells is preventing from our own immune system at the same time from harming our own body. And the memory T cells will get prepared to create a sort of a memory about this kind of an action or this kind of a war between the antigens and the antibodies, so that this data is kept for future, and hence we are able to toss out the antigens later when they come. However, the B lymphocytes, their mechanism is slightly different. B lymphocytes are sensitized directly by both the antigen as well as the helper T cells. So you all already know that the helper T cells are one of the clones of the T lymphocytes. And this is where the relation between the T helper cells comes with B lymphocytes. Now, the activated B lymphocytes multiplies very fast and produces a clone of plasma cells and memory B cells. The plasma cells then would produce specialized glycoproteins called as antibodies, and these antibodies will be circulated throughout the body fluid like blood and lymph. The antibodies may either bind to a cell membrane or they may remain free. Those who are free will perform three important actions. First is agglutination, where it is able to sort of capture or engulf or immobilize the entering pathogen, bacteria or virus, and this is done with the help of phagocytes. Then the opsonization, or it's the coating of the bacteria. This one is the phagocytosis mechanism done by the macrophages. And the third is the neutralization of the toxins that the bacteria will release. The bacteria releases, or the virus will release certain kind of chemicals within our body that can be harmful to us. So we need to neutralize or to remove such kind of toxin materials from our body. And this is the kind of mechanism both the B and the T lymphocytes perform. Next topic is vaccination. Administration of a vaccine to protect against any particular pathogen is called as vaccination. Now, the body's immune system is suitable to protect us from any pathogens that can cause an infection. It is a very efficient system, maximum amount of the times. However, there may be a certain time when these pathogens may overwhelm or be more stronger than our own immune system. So what happens like this? That is the time when we feel that we have got an illness and we need medication. So vaccinations are a way to teach our immune system how to recognize and eliminate the pathogenic organism. So it's basically preparing ourselves to deal with any kind of invasive pathogen. So therefore, vaccines, yes, they are very safe compared to drugs. Even I would say that vaccinations are very safe because they're directly administered into the blood. They do not undergo any kind of mechanisms in the other parts of our body. They will directly go to the required site. Yes, may not be 100% efficiency of the vaccination, but still it is way more than what a drug can offer. And however, all vaccinations are studied, examined on a large number of animals, and then eventually put to us. So therefore, research has been completed before it is administered to the general public. Next topic is the structure of the antibody. Antibodies are glycoproteins which are highly specific to specific antigens. We already know that. Our body makes only those antibodies which can attack a particular antigen. And otherwise, it will keep creating new ones for every new antigen it meets. It will create a new antibody for every new antigen it meets. Now the structure. The antibody is a Y-shaped molecule. Each immunoglobulin molecule is made up of 4 polypeptide chains. There are 2 heavy H chains. The ones which are in brown color, they are the heavy H chains. And we have 2 light or L chains, the ones in blue color. The 4 polypeptide chains are held together by the disulfide SS bond that give it a Y-shaped structure. You can see the disulfide bonds present between the yellow and the blue structural parts of this antibody. The region holding together arms and the stems of the antibody is termed as the hinge. You can look very carefully, there is a marking called as hinge. Then following, each chain of the antibody includes 2 different regions, the variable region and the constant region. Please look at the brown chain. There is a variable region of the heavy chain and there is a constant region of the heavy chain, both of different colorations. In the blue, there is a variable region of the light chain and a constant region of the light chain. The variable region constitutes the antigen binding site, also known as the paratope. It is this part of the antibody that recognizes and binds to a specific antigen to form the antigen antibody complex. Since most antibodies carry 2 antigens binding sites, they are said to be bivalent because it has 2 binding sites. Moving next. Our topic for the next one is the formation of the antigen antibody complex, and the study of the antigen antibody interactions is known as serology, another area of study. Combining sites of antigens are called as antigenic determinants, which react with the corresponding antigen binding site of the antibodies called as the paratopes, as I also specified in the structure. The antigen binding sites are located on variable regions of the antibody. Small variations in the variable region make the antibody specific to a particular antigen. The variable region enables the antibody to recognize the specific antigen and binds to a specific antigen in a lock and key manner, forming the antigen antibody complex. And that is how the antigen antibody complex mechanism works. The antigens on our blood cells. There are several known antigens on the surface of the human red blood cells. These antigens give rise to different groups, and there are many such genetically determined blood groups according to different mechanisms like the ABO, RH, Duffy, KID, Lewis, P, MNS, then the Bombay blood group, etc. So let's talk about first the ABO system. This one was developed by Landsteiner in 1902, and he was awarded the Nobel Prize for the discovery of the blood groups. He found 2 antigens on the surface of the human red blood cells and named them as antigen A and antigen B. He then noticed the corresponding antibodies in the serum called as small a and small b as the letters he will be giving. Now, the ABO system, in this, the blood groups are determined by the presence or the absence of an antigen A or B, and the blood group of a person is classified into 4 groups: A, B, O, and AB. Now, a person with blood group A, individuals with blood group A have the antigen A on the surface of the RBCs or the red blood cells, and the antibody is present in their plasma, which is antibody B. Similarly, for group B people, the antigen B is on the surface and the antibody A is in the plasma. For those who are in group A and B, they have no antibodies in the plasma, but they have antigens A and B on the surface of the red blood cells. However, people with blood group O have no antigens on the surface of the RBCs, but they have antibodies A and B both present in the plasma. Then there would be a question about what's the RH factor and what's the RH system of this whole ideology in the blood groups. The RH is the most complex of the blood group system. The RH factor is about an antigenic protein present on the surface of the red blood cells in human beings. First discovered by Landsteiner and Wiener in 1940 on the surface of the RBCs of the rhesus monkey. Hence, it is called as the RH factor, also called as the D antigen. Persons having the RH factor are called as RH positive, and those lacking this D antigen are called as Rh negative. Now, the Rh or the D antigen induces a strong immunogenic response when introduced into Rh negative individuals. The Rh blood group is an important factor in blood transfusion and involved in the hemolytic disease of the newborn children called as erythroblastosis fetalis. Basically, the destruction of the erythrocytes of the fetus. It occurs when the Rh negative mother conceives an Rh positive fetus. The Rh positive RBCs from the fetus may enter the mother's circulatory system during the childbirth, causing her to produce anti-Rh antibodies. As a result, subsequent Rh positive fetuses will be exposed to an anti-Rh antibodies produced by the mother, which results in, but obviously, death. The Rh negative mother is injected with the Rh antibody during all pregnancies carrying Rh positive fetuses. And this is about the Rh factor. So here I have kept a few questions for you all to think over in context to what we have just discussed during this whole video presentation. Go through them, put a little thought, and try to find the answers for yourself. Thank you. Please do subscribe, like, and share to our channel. Thank you. Your support will always help us create more educational videos for students. Thank you.